Okay, squad, let's look at Unit 5, Study Guide, page 2. Starting with number 4, describe how mitosis and meiosis are different. So these answers can be found on slide number 31, 32, 33, and 34 of the slideshow. The little device I give you for remembering this is if you pretend that we're like in a crossword puzzle. In mitosis, the I stands for identical. And in meiosis, crossword puzzle style, meiosis is going to create sex cells. So the E is for sex. It's not exactly accurate crossword puzzle work, but I think it gets the point across, and it's a way to remember it. When you see the I in mitosis, you think identical. And when we are talking about what does identical mean, if we have 46 cheek chromosomes in a cheek cell, a regular old somatic cell like your cheek, and you bite the inside of your cheek, that hurts. You need to repair that damage. It's going to go through the cell cycle, and it's going to make 2, hopefully, identical cheek cells, each with 46 chromosomes in them, because 46 is identical to 46. So mitosis creates 2 identical diploid somatic cells. All of the cells in your body are diploid except for sperm or eggs. That's what these guys are. And these guys, these gametes, sperm or eggs, come from meiosis, the creation of sex cells. So you have 46 chromosomes in a somatic cell, goes through meiosis 1. What also occurs during meiosis 1, which is really important for genetic diversity, is there's a little bit of crossing over that happens. And if you remember, crossing over is when the chromosomes kind of, rub up on each other and they swap genetic information. So if you have a green and a brown, after crossing over is over, you have some that have a little bit of brown on them and a little bit of green on them. They've shared genetic information just to scramble things up a little bit. So crossing over only occurs in meiosis 1, does not happen during mitosis. Identical. Meiosis 1, crossing over occurs, 46 chromosomes. Meiosis 2, the reduction division. Now we have 23 we have 23 chromosomes in 4 genetically different, it's an N, genetically different haploid sex cells. And remember, the way I remember haploid, I think half. Diploid, I think double. Again, not accurate, but it helps me remember. And these are going to be making sperm or eggs. So how are mitosis and meiosis different? This is how. What occurs during the 3 stages of interphase? My artistic ability is coming through right here. You've got G1, S, and G2. G1 stands for gap 1. This is where your cell is just being cellular. A little bit of growth happens because, it doesn't just infinitesimally get smaller after each division. So there's a little bit of growth, but it's mostly when your cell is just being cellular. S stands for synthesis. And what are we synthesizing? We are synthesizing DNA. So it's the creation of new DNA, DNA synthesis. The old DNA is used as a template to make identical strands of new DNA, because remember, we want them to be identical. Everything should be the same. And then G2 is gap 2. And this is where there's enzymes that are kind of they're looking for mutations. Enzymes checking everything over. They look for mutations. They try to make sure everything is correct and good to go before we go into mitosis. So we're going in this direction. Okay, so this is interphase G1, S, and G2. And this is what we do before we get into prophase, metaphase, anaphase, and telophase. Let's take a look at the slideshow really quick. On the slideshow, this can be found in here. Okay, G1, growth, GS, DNA replication, G2, mitosis, and then that's interphase. This picture's a little bit better than the one I drew. Same thing, G1S, G2, and then here's another picture of this for you. G1, S, G2. G2 double checks the duplicated chromosomes for errors, making any needed repair. And then this is the picture of synthesis. You've got your original strand of DNA. Now you have your two new daughter strands using the original strand as a template for both. All right, back to the study guide. Number 6, more artwork. Yes, Pamela should have been an art major. No, false. Okay, prophase is when we have our cell, there's our cell membrane, there's your nuclear envelope starting to disappear. Your centrioles are going to move to opposite poles of the cell. They're going to help pull the cellular contents apart when it's time. Asters and spindle fibers are little filamentous type structures that start collecting here. And the chromosome, the DNA is going to condense and start to form chromosomes. So that's what's happening during prophase. Let's go over to the slideshow and take a look at prophase. I really like this graphic that shows how the DNA starts to condense and it forms the chromosomes. So when we look here, remember your DNA was just kind of like hanging out loose, disorganized in the nucleus. And then when prophase comes along, it starts to organize. It starts to wrap itself around little proteins called histones. And then it's going to supercoil and wrap itself up some more and become these really nice organized chromosomes. So that's slide number 26 and 27, showing how the DNA forms into chromosomes during prophase. Sliding on down here, there's basically the same picture I drew. Okay, there's your prophase. This is prophase under the microscope. You can see the condensed, it's now chromatin on the inside, the condensed DNA. All right, next, metaphase. Easy to remember because M makes me think of middle. In metaphase, you're going to have your chromosomes lined up along the equator of the cell. I only drew 5 here, but if it was us, it would be 46 of them. And if you're ever getting some genetic testing done or you're having somebody go and look at your genes, they are going to try to find one of your cells in metaphase because this is as organized as they're going to get. So looks good. Again, back to the slideshow. Like I said, it's basically the picture I drew. This is using a couple different types of microscopy. Some have some radiated images where someone has maybe consumed something that causes their chromosomes to glow, like a radioactive substance that will make your chromosomes light up under certain types of microscopy. So pretty cool stuff, I think. Anaphase is our next stage of the cell cycle. Our next stage of mitosis, nuclear division. Back to the study guide. There we go. There you go. Anaphase. The chromosomes are now being pulled apart by those esters and spindle fibers, and they're starting to migrate to opposite sides. They're pulling apart. Anaphase is really important because if the breakup doesn't happen properly, your new cells are going to have the wrong amount of genetic information. So if we're a human, if this was a human cell, we would want 46 chromosomes heading in this direction and 46 chromosomes heading in that direction. We don't want the same amount. We don't want different amounts. We don't want 45 and 47. We would have a problem there. That's called non-disjunction. If any of these chromosomes fail to break up during anaphase, they try to keep texting. They're just going to be friends. You know that doesn't work out, and biology should teach us that. Let the breakup be nice and clean, a good clean split. No non-disjunction, because then you end up with the wrong amount of chromosomes, and this is where some genetic disorders can crop up. We're going to talk a lot more about this in Unit 6 and 7, but just remember me telling you how important the breakup is, how important the anaphase is. Okay? More pictures from the slideshow. Basically the same thing. This is a cool picture under the microscope. This is an onion cell of the root of an onion, and you can clearly see that breakup, that divide that occurs during anaphase. There she goes. Okay, we want a nice clean break. Back to the study guide. Telophase is when the cell takes a little bit of an orbital peanutty shape, because we're starting to get all these chromosomes now are going to collect together into new clearly defined nuclei. So you start to see this dark region where the new nucleus is beginning to form. Back to the slideshow. There's our telophase. Pretty cool. That's a nice image from the onion root. This little picture of telophase. The nuclear envelope is starting to reform. You've got the chromosomes all tucked in where they're supposed to be. And we're starting to see the division of the cytoplasm, which is our next step. Not technically, that's why I put that asterisk there. Cytokinesis is part of the M phase. It's part of the M phase of the cell cycle. That includes all of this. But it's not technically part of mitosis. Mitosis is, officially speaking, just nuclear division. But cytokinesis gets lumped into the M phase, and that is the division of the cytoplasm. So how is cell division different in plants and animals? Just embarrassing words for your biology teacher to have to say. In plants, you've got your nice cell plate. That makes so much sense. It's this little region of cells that forms in between so the cell can nicely divide. In animals, it's a cleavage furrow. And that's what that looks like. Back to the slideshow. Yep. There she is. Great. And then cell plate. Cell plate, that's what happens in plants. Little vesicles form. We're going to start to divide the cytoplasm so you can get some two new cells. There you can see that occurring in our onion root. Nice, clean cytokinesis. And the way I remember cytokinesis is it's the division of the cytoplasm. When you were defining your vocabulary terms, hopefully you made that connection. Cytokinesis, cytoplasm divides. All right, that does it for page 2, and I will see you in a few minutes for page 3.